This week in Topic 3, we're going to be looking at microbial metabolism and the genetics. This is kind of a long topic with a lot of details, so I'm going to split it into two videos. For this first one, we're going to be focusing on the microbial metabolism, so this is going to be Chapter 6 in the textbook. Our objective is going to be the first one for this week. Describe the central metabolic pathways in the microbial world. We'll address the other three objectives in the next video. So again, we're starting with Chapter 6, Microbial Metabolism, and we're going to be focusing on sections 6.1 through 6.5. So first off, we want to identify what is a metabolism. So this is kind of a big idea of just how organisms can use energy and molecules. So if we're building things up or breaking things down, that's all metabolism. So the two main parts of it are going to be catabolism and anabolism. Catabolism is when you break down molecules in order to free up energy to be used. So whenever you think about cats, they like to tear things up and break things down. So you can associate catabolism with breaking things down. With anabolism, that's going to start with smaller molecules and use energy to produce larger molecules. So in our little graphic here, we see that we have catabolism, that's our energy source of glucose, and it can produce energy. And the waste products, such as carbon dioxide and acids, are going to leave a cell. Anabolism is going to be where you start with smaller molecules or smaller elements and add energy in there in order to build up into larger structures, eventually making like the cell wall. So now that we've talked a little bit about this, we really need to understand the concept of energy. So energy is kind of one of those words that gets thrown around a lot and can mean a few different things depending on who you ask. But we're going to look at energy in kind of a science form, but I'm going to try to simplify it as much as we can. So energy is the capacity to do work. What is work? Well, I just would call it stuff. Energy gives something the ability to do something. So what does that mean? Let's look at if you're in a house with a ceiling fan. Energy is going to go into that fan and allow the blades to spin. So energy is allowing the fan to do work. So energy cannot be created nor can it be destroyed. And energy can be converted from one form to another. So this is a great example here with this hydroelectric dam. So in this video, or in this picture, we have four different, well, really we can only see two of those energy types, but we can infer two more. So we have potential energy and kinetic energy. The potential energy is the ability for something to do something. So in this case, that would be the water is held up by this dam. Normally, the water wants to just flow downhill and seek the lowest place possible due to gravity. So that's potential energy. Well, it gets converted into kinetic energy when they open up the turbines and allow the water to flow down. Well, they can capture some of that kinetic energy with the turbines and use it to generate electrical energy. And down the line, that electrical energy can go and charge up your cell phone battery, and that would be chemical energy. So energy is just going to be the ability to do something, and it can be transferred. Other types of energy we see here would be like sunlight, and sunlight is actually going to be where most energy on Earth comes from. As a matter of fact, this water could not have gotten up above the dam unless the water had evaporated due to sunlight exciting the water molecules, and eventually it would have rained upstream, and that would be how the dam gets filled. Okay, so sunlight is the energy source for most of life. Some forms of life can use chemical energy, but for most cases, sunlight is the energy for light. So photosynthetic organisms are able to harvest the sunlight and use photosynthesis to create higher energy, larger molecules. Now remember, that would be anabolism because we're building up. And whenever we're consuming energy, this kind of a reaction would be called endergonic, because the energy is being pulled into the chemical reaction. Now, going down the line, mice, or going up the line, however you want to describe it, mice are going to be able to eat the grains and break down those compounds, those molecules, for energy. So heterotrophs will consume them and release energy. And remember, if we're breaking something down and releasing energy, that is catabolism. And if we're releasing energy, the energy is exiting the chemical reaction, so we would call that exergonic. Okay, so now let's go ahead and look at kind of the framework of metabolic pathways. So the big picture, you're going to start with some sort of starting substrate and end with an end product. And there's a few different pathways that can make this possible, and a few different major things. But first thing that I want you to notice is that we have different steps with our pathways. Well, why can't we just go from our starting substrate to the end product? Well, that has a good answer. That's because, let's take the idea of glucose. So if we take glucose or sugar and we are able to oxidize it or release the energy by lighting it on fire. So if we light glucose on fire, all the energy stored in those molecules is released. But it's not very controlled and it could be dangerous. So what organisms are going to do is we're going to use intermediates. This will control the reaction and control how much energy is released at a time. And also, if we're building something up, then it requires less energy to go through each intermediate. So it can make this go forward a little bit more effectively and more controlled. We also use enzymes in between each intermediate. Enzymes just make reactions occur rapidly and effectively, and we're going to talk more about enzymes later on. So we have three kind of major motifs or major pathways that metabolic pathways can have. So either it'll be linear, so that's just you start with A and end with B, or you can go through a branched one where you start with A and it can be broken down into end product 1 and end product 2. And then you can have cyclical, where you have a starting substrate and it generates intermediates that will eventually interact with that starting substrate but produce the end product as a result. And we're going to see all three of these pathways today when we're looking at the central pathways. So I said we were going to talk about enzymes, and so here we are. Enzymes are going to be what we call biological catalysts. They're going to be usually proteins. So what is a catalyst? Well, a catalyst is just something that causes a reaction to happen more quickly or easily. Catalysts do not get consumed in a reaction, so they will be there to speed up other reactions as well. So the way that they speed them up is by making reactions more favorable, like by lowering the activation energy. So if we look at this graph, we have the relative energy required for each of these states. So in order for a chemical reaction to occur, we're going to need our products to have a small investment of energy. So the blue line tracks the energy required without enzymes. It requires a lot of energy invested in order to have the products be formed. So by moving up, that consumes some energy to start, but then it moves down, so it frees up that energy again. So this actually slows down chemical reactions from occurring because it requires so much more energy. But if we have an enzyme, we will follow the red line. We don't need to get as much energy invested in before we get that payoff. It's much quicker and requires less energy. So with this in mind, chemical reactions can actually occur, like the ones that we need for our body to exist. Those reactions happen, it's just they are way too slow to sustain life. But with enzymes, it gets sped up and then life can exist. So one last thing before we get into our pathways is going to be the idea of adenosine triphosphate, or ATP for short. So ATP is what we call the energy currency of cells. This is just going to be the way that energy is usable in the body, and it can be sent all around. But it's created in catabolic pathways. Remember, catabolic means we're breaking things down to release energy. So adenosine triphosphate is created in these catabolic pathways in a process called phosphorylation. So we have two different kinds of phosphorylation. We have something called substrate level phosphorylation and then oxidative phosphorylation. We'll come back to those terms later, but just know that there's two different types of phosphorylation. So what is phosphorylation? This is going to be where we take a phosphate and we're going to bind it to an ADP. So ADP means adenosine diphosphate, and once we bind the third one, it's adenosine triphosphate. Now each of these bonds between phosphates is high energy and they're pretty unstable, so it's pretty easy to release the phosphate and also release the energy. So ATP will convert back to ADP when the energy is released, just waiting to be recharged. And if we really need a lot of energy, we can also break down ADP and release one more phosphate and make AMP, which would be adenosine monophosphate. But yeah, this is just how we're able to transfer energy around the body or around an organism and have it be easy to release. Okay, so how does chemical energy get released? Well, we need to cover something called electronegativity. So electronegativity is just how much an atom or molecule wants to grab electrons. So if you have a chemical with a high electronegativity and it has an electron, there's pretty low energy required for that to exist. But if we have a low electronegativity, grabbing an electron, that one actually has a lot of energy invested. So if you have a higher electronegativity take an electron from a low electronegativity molecule, that actually is going to release a lot of energy. And so that's what we're seeing here. When we have compound X transfer its electron to compound Y, then we're going to release a lot of energy because compound Y really wanted that electron. And compound X had so much energy because it was holding the electron, it's like, I don't want this. So this is what we're going to call a redox reaction, or a reduction-oxidation reaction. So oxidation is a loss of electrons. So if a molecule loses electrons, it's being oxidized. If a molecule is gaining electrons, it's going to be reduced. And the way that you can remember that is oil rig. So oxidation is loss, and then reduction is gain, oil rig. So if you're losing an electron, that's oxidation. If you're gaining it, it's reduction. And then it can get a little bit more confusing when we're talking about the reducing agent. It's the thing that's causing something to be reduced. So the reducing agent would be what's being oxidized. But you don't need to really get into that too much. So hydrogen or protons are typically going to follow electrons in cells. So if you have an electron being moved from one compound to another, a hydrogen is going to typically follow it. It's just because electrons have that negative charge and hydrogen ions or protons have a positive charge, so they just really want to follow it. And so redox reactions inside of organic organisms are typically going to have a terminal electron acceptor, or TEA, and that is what ends up with the electron at the very end of a metabolic pathway. Okay, so energy release. Energy is going to be released when an electron is moved from an energy source to a terminal electron acceptor. So let's go ahead and look at this image right here. So we have glucose, and this is a high energy molecule. This is also known, you might know this as sugar. It's kind of like table sugar. Well, kind of. But anyway, that glucose has a lot of energy, and it wants to release that energy. So we're going to release this by releasing or moving electrons. So depending on how many electrons are being released, tells us how much or how electronegative the difference is tells us how much energy is being released. So if we have glucose just releasing its electrons to pyruvate, this would be in like fermentation, you wouldn't have too much energy being produced. But if you have glucose sending its electrons over to oxygen, oxygen really wants electrons, so a lot of energy is going to be released in this case. So really, the energy just released depends on the amount of, or the difference between the two. So glucose releasing to pyruvate only releases a little bit of energy, but glucose releasing to oxygen releases a lot, and you might see this ammonia right here, or nitrate to nitrite. Sorry, not ammonia. But this is going to be a different pathway. This would be like anaerobic respiration, but we'll come back to that later. So, electron carriers, these are going to be really important things in your body. Electrons are not going to be released from glucose or other molecules being broken down all at once. Electrons will usually not go directly to a terminal electron acceptor. So for example, I talked about glucose and oxygen. Oxygen would be the terminal electron acceptor. So if we had the electrons going straight from glucose to oxygen, that's would actually be fire. So if you've ever lit sugar on fire, that would be going straight to the electron acceptor, and that can't happen in organisms. So instead, we're going to use something called electron carriers. So electron carriers are just these little organic molecules. Well, not organic. They're just these little, maybe they are organic, I don't remember. These are little molecules that like to hold on to electrons, and they'll transfer them from one place to another. If they have an electron, they have the ability to reduce something else. So we have a few different types, and they have a longer name, but typically they'll be called like NAD, FAD, or NADP. That's in their oxidized forms when they don't have electrons. But when they are reduced, they will gain an electron, and they'll be called NADH, FADH2, or NADPH. So all of that just means is that these different electron carriers will gain a hydrogen because hydrogens follow the electrons. Just to go over the names, we have nicotinamide adenine dinucleotide, that's NAD, or flavin adenine dinucleotide, FAD, or nicotinamide adenine dinucleotide phosphate, NADP. These are going to be generated or be used at different points of metabolic pathways in order to move electrons. There are a limited number of electron carriers within an organism, so that will be important to understand later on as well. Okay, another thing before we get into metabolic pathways are precursor metabolites. So precursor metabolites are just intermediates that can be used to make other biomolecules. So these catabolic pathways can be linked to anabolic pathways just because of precursor metabolites. So as we're breaking something down in a catabolic pathway, we might create a precursor metabolite, and that can be used to build up a different pathway in anabolism. If biomolecules cannot be made in a cell via precursor metabolites, we would actually need to obtain those from the environment. So, for example, in humans, we have no way of generating some amino acids. So we have to obtain those from our diet. An example of that would be like methionine. You can't make that yourself, so you have to eat it. But in bacteria, they have different things that they can't make. So let's look at some of the precursor metabolites. So glucose 6-phosphate, that's going to be used in glycolysis, but we can use that to make lipopolysaccharides in cells. So that's just going to be the substance on the outer layer of the cell wall, if you remember that from topic 1. Another one that we can use is ribose 5-phosphate. That can be used to generate nucleotides, but also proteins. And that's going to be created in the pentose phosphate cycle. Anyway, we're going to move on. So glucose is going to have a lot of purposes, but the main pathway we're going to be looking at is the pathway used for the release of energy, and that's called catabolism. But it can exit the pathway and be used for biosynthesis in anabolism, and that's going to be the idea of precursor metabolites. So our glucose molecules are going to go down the main pathway, which will eventually create carbon dioxide and energy. But it could also branch off to create lipids. So that's going to be things like triglycerides, phospholipids, anything that, it could also branch off to create amino acids if the cell needs. It can branch off to make carbohydrates, different types of carbohydrates, or it could branch off to make nucleic acids. So the central metabolic pathways are going to be like 3 pathways that completely oxidize glucose to release energy. So our pathways are going to be glycolysis right here. Well, that wasn't quite a star. Our pentose phosphate pathway up here, and then our TCA cycle, you might also know it as the Krebs cycle or the citric acid cycle down here. And then those will all channel, those will break down the molecules down into carbon dioxide, but eventually the electrons will be used in cellular respiration down here. And that will use the proton motive force to phosphorylate more ADP. But we'll get to that later. So the main idea of each of these is that we're generating ATP. So anything in this box is going to be, we'll make it this box right here. This one right here. Anything, here, let me clear the screen and just make that better. Glycolysis and the TCA cycle are going to be substrate level phosphorylation. This is because as the substrates are being broken down, phosphorylation is occurring. Down here in cellular respiration, that's going to be called oxidative phosphorylation. That's because we're using redox reactions or oxidation reduction reactions to phosphorylate. And we'll talk more about that later as well. And then fermentation. This is going to be when respiration is not able to occur. So that means you don't really have a great terminal electron acceptor. So electrons from the glucose will need somewhere to go. We have a very limited number of, well, not a very limited, but we do have a limited number of electron carriers. So we can't have those busy carrying electrons from glucose. So what we're going to do is use pyruvate, which is a slightly oxidized form of glucose, and turn that into an electron acceptor. And we'll talk more about fermentation later on. Just know that fermentation will only release a small amount of energy because it breaks off too quickly. And aerobic respiration or respiration will generate a lot more energy. Okay, so now we're finally moved on to section 6.2. We're going to be looking at enzymes. So enzymes have a few major things. So first off, let's look at it. We have the enzyme, that's this big blue blob right here. And then we have something called an active site. The active site is going to be where enzymes can interact with substrates and catalyze reactions. So what we have here is this little pink or purple substrate binding with the active site of the enzyme. This creates the enzyme substrate complex. When this is formed, the chemical reaction is being catalyzed, and then once it's completed, the enzyme will be released unchanged, and the products will be released and they are changed. So just remember that in an enzyme catalyzed reaction, the enzyme will not be consumed. The enzyme will still be there in order to help another reaction be catalyzed in the future. But it just speeds up how the reaction can occur. Okay, enzymes may need cofactors in order to function. Cofactors are non-protein substances needed to speed up reactions. So a couple examples would be like copper, zinc, or magnesium. Those are all things that you need to have reactions occur with different enzymes. Another type of cofactor is called a coenzyme. This one's going to be an organic molecule, so this would be like the NAD, the FAD, or the NADP. But another one that you might know about is ascorbate, also known as vitamin C. This is a cofactor needed, and if you don't get it, you could develop scurvy as a human. So enzymes may not be able to function without their cofactors or coenzymes. Coenzymes and cofactors can participate with a lot more different reactions than a single enzyme has. So we have many different types of enzymes because they're usually specific for one, maybe two reactions, but cofactors and coenzymes can be used for pretty much anything that they need. Not, they can't be used for everything, but yeah, they're much more versatile than enzymes. So some more examples of coenzymes, we have folate or folic acid. Um, we have niacin, that creates NAD. Pantothenic acid, that's coenzyme A. Pyroxidine, that's vitamin B6. Riboflavin B2. And then thiamine, that's vitamin B1. And as you can see, some of these sound familiar, FAD, NAD. Yeah. Okay, and then environmental factors, how can they affect enzymes? We covered this a little bit with the ideal conditions for bacteria to grow in, but this is really where we can see it. Enzymes can be affected by an environment. So like if they don't have enough water, enzymes can't exactly interact with the molecules they need because usually you have water help move things around. Another one is pH. So the acidity, if we are not within the optimal pH, the effectiveness of a protein can drop off quite rapidly. So if you're in an acidic environment but you're more basic or more neutral, that enzyme's not going to function very well. And then another one is temperature. If it gets too cold, the enzyme might change shape a little bit and not be able to fit the substrate. On the other hand, if it gets too hot, the enzyme might completely denature and unravel, and it won't function at a high temperature either. Another way that enzymes can be affected is by something called allosteric regulation. Allosteric just means elsewhere, basically. So allosteric regulation means that it's being regulated by something binding outside of the active site. Allosteric regulation may increase or decrease the effectiveness of an enzyme. So sometimes having an allosteric activator could make the reaction happen quicker, or in the example here, an allosteric inhibitor can slow down a reaction. Something to take note of with allosteric regulations is that they are always reversible. Your body wants to be able to slow down or speed up a reaction, but it never wants to stop it, and same thing with bacteria. They don't want to stop it dead in its tracks, they just want to modulate it, speed it up or slow it down. So we have different inhibitors. We'll look at a few. First one would be a competitive inhibitor. We'll take more time and look at that. We have non-competitive inhibition, and that'll be by regulatory molecules. We'll look at that too. But that one's going to be reversible. And then non-competitive inhibition by enzymes, and those ones are going to be poisons. They're not reversible and can become problematic. So competitive inhibition, this is when an inhibitor is going to bind with the active site of an enzyme. So the inhibitor is going to have a similar structure to the substrate. So in this picture, we have sulfa drugs, which are used to as an antibiotic to kill off E. coli, and that's because E. coli cannot consume folate, so it has to synthesize it, whereas your body can consume folate. You don't need to synthesize it. So what we can do with the sulfa drug is give you the sulfa drug, and the sulfa is going to bind to the enzyme that the bacteria uses to synthesize folate, and then it'll get stuck in that enzyme, and the E. coli can die off. This won't affect you because your body can use environmental folate. So non-competitive inhibitors, they do not bind to the active site. That's why they are not competitive, because they're not fighting with the substrate. They can change the shape of the enzyme. So in this example, the active site gets deformed once the non-competitive inhibitor binds. Again, Non-competitive inhibitors can be a form of allosteric inhibition. However, sometimes they can be due to a toxin or poison. Okay, next we're going to look more at the central metabolic pathways. So glycolysis is going to be one of those big ones that you hear about. So this is just a series of steps used to break down glucose. We're going to start up here with a single 6-carbon structure. So each of those gray balls represents a carbon. And what we're going to do is we're going to process this carbon. So we're going to add a phosphate to it, make it glucose 6 phosphate, and then we're going to rearrange it, make it fructose 6 phosphate, add another another phosphate, make it fructose 1,6 bisphosphate, and then we're going to split it up into 2 3-carbon molecules or structures, and one of them is going to be made directly into glycerol aldehyde 3-phosphate, and the other one will go into an intermediate called dihydroxyacetone phosphate, but eventually it will be converted into G3P as well. And again, we're going to keep processing these, and you'll see we generate NADH, and then we'll also generate ATP here, and then ATP here. So in the end, we're going to invest 2 ATP, but we'll get 4 out. So that's a net gain of 2 ATP. And we're also going to generate 2 NADHs. And those can be, the ATP is going to be the substrate level phosphorylation, and we can use those electrons later on for oxidative phosphorylation. There's also going to be 6 precursor metabolites within glycolysis. That's going to be glucose 6-phosphate, fructose 6-phosphate, dihydroxyacetone phosphate, 3-phosphate, phosphoglycerate, phosphoenol pyruvate, and then pyruvate. All those things can be used in other pathways to build up biomolecules. Okay, the pentose phosphate pathway. This is going to start with glucose as well. But this one's not actually going to create ATP directly. Instead, you're going to make a few different things. This one can produce glyceraldehyde 3-phosphate, or G3P. That might sound familiar because that is something that we can use in glycolysis. But something more important is that we can use this to create molecules important for biosynthesis. One of the most important ones is ribose 5-phosphate. So ribose might sound familiar to you if you remember RNA, because ribonucleic acids use a ribose sugar in their structure. So this can help us create nucleic acids. As well, this can be used to generate NADPH, which can help us create other biomolecules as well. So the pentose phosphate pathway doesn't create ATP itself, but it's used in biosynthesis of other molecules, and it can send G3P back into the glycolysis pathway. All right, the next one is going to be the tricarboxylic acid cycle, or you might also know it as the Krebs cycle, or even the citric acid cycle. So where does it get its name from? Well, Krebs is going to be from the scientist. Tricarboxylic acid comes from it starting with 3 carboxylic acids bound together. Boom, boom, boom. Or you might know it as citric acid cycle because citrate is going to be like citric acid. Okay, cool. So starting off with pyruvate, pyruvate is going to be generated through glycolysis, and it's the last part of glycolysis. Well, this isn't quite ready to go into the citric acid cycle, so instead we're going to send it through a transition step first. So this is going to be used to convert pyruvate into acetyl-CoA. So in this, we're going to oxidize it and release a carbon dioxide, and we're also going to generate an NADH. This will be used for later. But once we get to this point, we will have our acetyl-CoA. Acetyl is going to be that 2-carbon molecule, CoA is the carrier. So once we send this into our Krebs cycle, the CoA will go and be released, and the acetyl group is going to bind to this oxaloacetate to make citrate. Well, I'm not going to go through all of the steps like that, but just know that we're going to generate NADH right here, and then we're also going to generate NADH here. And as you can see, we're also releasing carbon dioxide. So this means that we're really oxidizing what we started with, because we're removing basically all of the higher energy electrons. And then eventually we're going to generate some more, well, this says ATP, but if I remember correctly from my biochemistry classes, it's actually GTP, but that's a concept that you don't really need to explore too much. And then we'll also create some FADH and then some more NADH. So in this, we're really releasing a lot of electrons, releasing more energy, but we're also moving those electrons to an area we'll be able to use later. So this is going to generate 2 molecules of ATP, and then it's going to generate 6 molecules of NADH and 2 molecules of FADH2. So again, here's a summary. It goes over everything, just the base, and dilutes it down to what it creates. And yeah, well, you can pause on this part if you need to read any of it, but I'm not reviewing it in this video. Okay, next up is going to be cellular respiration. So cellular respiration starts off with the electron transport chain and creating a proton motive force. So the electron transport chain is a series of membrane-bound it requires a series of membrane-bound electron carrier proteins. Each of these carriers have a greater electronegativity or electron affinity than the last. And so you can imagine this as dropping a ball down the stairs. So dropping the ball wants to go to this stair, but then the next step wants it more, so the ball will drop. So this is going to be like each of those electron carriers in tandem. Each one wants the electron more than the last. So just like the ball falling down the steps, eventually it'll hit the ground floor, and in this case it'll be oxygen. But as this happens, it will create an electrochemical gradient, and this will cause hydrogens to be pumped through to the other side, and this will create something called a proton motive force. We'll come back to this later. But note that the electron transport chain and the proton motive force are going to be generated in the mitochondria of eukaryotic cells. Now remember, eukaryotic cells have organelles specially functioning to do this, but in bacteria, they don't have organelles. They're a little less organized, so in bacteria, it'll happen in the cytoplasmic membrane. Any prokaryote, really. Okay, so the electron transport chain in the mitochondria, this is going to be a lot to take in, but in the end, we have 4 different complexes that we're going to be looking at, and then a final pump called ATP synthase. So let's start off with complex 1. This is also known as NADH dehydrogenase complex. So this is right here at the beginning. So what this is going to do is it's going to accept the electrons from NADH. And when it does this, it will pump 4 protons across the membrane. So in this, inside the mitochondria, it's going to be using those cisternae, or cristae, not cisternae, cristae. And we're going to have the 2 membranes and then the intermembrane space. So we're pumping 4 hydrogens into the intermembrane space. And that electron will eventually be passed off to a ubiquinone, and that'll be passed off to complex 3. Now we'll come back to complex 3. Now let's look at complex 2. Complex 2 is also going to be known as succinate dehydrogenase complex. This one can accept electrons from FADH2. This one, however, is not a transmembrane protein. It will only accept electrons. It will not pump across any hydrogens. but it will send those electrons with the ubiquinone, and that will move over to complex 3. So now we'll talk about complex 3. In this, we're going to have the electrons travel from the ubiquinone in through complex 3 and eventually be transferred to cytochrome C. When this happens, 4 protons are going to be pumped across the membrane into the intermembrane space. Note that NADH will move a total of 8 protons across, whereas if we start at complex 2 with FAD2, we're only moving the 4 right here, plus 2 more later on. Okay, and then complex 4 is also known as cytochrome C oxidase complex. This is going to accept the electrons that have been transferred to cytochrome C, and so these are going to cause 2 more protons to be pumped across the membrane. And then complex 4 will eventually move the electrons back into the mitochondrial matrix and have oxygen accept those electrons. Oxygen is also known as the terminal electron acceptor, and that'll generate water, H2O. So now we have those electrons got transferred and they're back, but the hydrogen molecules got stuck on this intermembrane space. Well, now this has created something called the proton motive force. Those protons really want to get back across the membrane because right now it's looking a little positive up here and they need some negativity. So those protons want to move back down their electrochemical gradient. Well, luckily, we have something called ATP synthase. Now this is going to have 2 parts. We have a channel and something that works kind of like a water mill. So the hydrogen molecules will be able to move through the channel of ATP synthase, and then this little water mill looking thing is going to spin and it's going to phosphorylate these ATPs with, or phosphorylate the ADPs to create ATP. So for every 10 protons that move through, we're going to get 3 ATP. And so this is how the electron transport chain will help create this proton motive force. And this will, the proton motive force is what's driving oxidative phosphorylation. We call it oxidative because we had oxidation happen over here. And then phosphorylation because we're phosphorylating ADP over here. So this also happens inside of prokaryotes, except they don't have mitochondria, so this is actually going to happen on their cytoplasmic membrane. So we have our extracellular space here and our intracellular space here, or the cytoplasm. So we have 2 NADH dehydrogenases, and those are actually going to be equivalent to complex 1. Okay. We also have another molecule that's functionally equivalent to complex 2. However, we do not have a complex 3 analog, but we do have a complex 4 analog over here, ubiquinol oxidase. So what happens is since we have one less step, we might not generate as much or move as many protons. Or actually, it's not just that we might not, we will not. Bacteria are less efficient because they don't have those organelles. But they will be able to move hydrogen outside of the cytoplasm, and the hydrogen will want to move back in. So it's still making that proton motive force. So we do still have ATP synthase, and that will have 10 hydrogens cause it to spin to create 3 ATP. However, in prokaryotes, they have other functions for the hydrogen molecules that are outside. So you might have active transport happening, so we can move a molecule along with hydrogen back into the cell, or we can have a hydrogen move in and cause a flagella to rotate. Now remember from topic 1, flagella help a cell move. Okay, so we also have something called anaerobic respiration. So this is still cellular respiration, but just know it's not as efficient as aerobic respiration. Anaerobic respiration will not use oxygen as a terminal electron acceptor. So because it's not using oxygen, there's actually going to be less energy released. So it will use other electronegative molecules other than oxygen. It's just they're not efficient. And because it's not using oxygen, it's called anaerobic respiration. Okay, another topic that you need to keep in mind when we're talking about theoretical yields is that experiments that were used to do this were done with rat mitochondria. So the results gave us an approximate 2.5 ATP made per NADH and 1.5 from FADH2. So because it was they didn't get exactly a whole number every time because experiments will rarely ever give you a perfect number. We had to use math and averaged out to 2.5 and 1.5. So in this textbook, and what we're going to do is round it up to whole numbers. So we're going to say 3 ATP made for NADH and 2 for FADH2. So this means that we're going to have a high theoretical maximum, but we might not actually achieve that because we have variations in the ATP generated. This plus the idea that prokaryotes can use the hydrogen, the proton motive force for other things other than ATP generation, we can see how we might not actually achieve those theoretical yields, both mathematically, like the experimental value will not meet the theoretical, but also we can see that the proton motive force might be used for something else. Okay, moving on to our last topic is fermentation. So again, sometimes we might not have a terminal electron acceptor present. So for example, we might not have oxygen available. So imagine if you're running and you start working out really fast. At first, you won't have enough respiration to keep up, or enough oxygen, so you won't be able to do your respiration. So you're going to be relying only on glycolysis. So remember up here, right here we have glycolysis, and we're going too fast to produce to get that oxygen going right away. So we're going to have fermentation going on. So with this, we're going to be generating pyruvate, but we don't have a terminal electron acceptor. But what we can do is convert pyruvate into something that can hold on to electrons for us. This way we can free up our electron acceptors to do other jobs that might be more important than holding on to the electrons from glycolysis. So two major pathways we have with fermentation is lactic acid fermentation. This is what we do as humans. And then we also have ethanol fermentation. This is what makes alcohol. And so basically what's happening is pyruvate is gaining an electron, so it becomes lactate. Or pyruvate is going to be converted to acetaldehyde, and then into ethanol. But we do really like fermentation. Fermentation is going to be, one, evidence that metabolism is happening, but two, it's going to make us our products that we really like with fermentation. So we can use lactic acid fermentation to make great things. We can make yogurt, we can make pickles, we can use ethanol fermentation or alcoholic fermentation to make things like wine and beer. We can use it to make cheese. So all of these things are going to be great for us. We're using some metabolic pathways that do not have a terminal electron acceptor. This is the end of topic 1. This is the end of section 6. In the next video, we're going to cover topics about mitochondrial metabolism, microbial metabolism and genetics, and those will be chapters 7 and 8 in the textbook. So that will be in the next video.